Hello and welcome to the University of St Andrews online events programme. I am delighted to have been asked to deliver one of the series of lectures for alumni, supporters and friends of the university, and I would like to thank you all for being here with me today. My name is Kishan Dholakia. I'm a professor of physics at St Andrews in the School of Physics and Astronomy, and my speciality is the science of light. Everything to do with light, from fundamental advances right through particularly to biological discovery, healthcare and other areas. And that's what I'd like to do. So the title of my talk today is Light for Life, New Advances for Healthcare, Biomedical Discovery and Beyond. The science and application of light has been around for hundreds of years. And today, light is an integral part of our everyday living and quite simply has revolutionized so many things we do. It's revolutionizing the way we run and power cities. We would do education for children and adults, and also make the most exquisite and important measurements that we can. For example, in the bottom right, you see the idea of a pulse oximeter, and that's a device that can measure the oxygenation of your blood and is used by hospitals all around the world. This idea of the science of light is very, very important because light holds a privileged place in the universe. It's very exciting. The speed of light is one of the constants that we know and a very important one, and it really underpins everything around us in everyday life. So today I'm going to describe a few of the recent reasons about why light's exciting, and then we'll move on to some of the work that we're doing at St Andrews. Here's a picture of the world from space, and astronomers often look for light pollution, and economists see the lights on Earth as a proxy for gross domestic product. But it turns out 1.3 billion people do not have access to electric light, with about half that again only having intermittent access. Lighting and the absence of it and the poverty is a major concern around the world. You can see here in Western Europe, United States, India, some parts of China and Australia, we have bright pockets of light, but the many parts of the world are not covered. An incredible picture that just brings home how much we can at the moment light up the world and how much more we need to do. The science of light is often known as photonics, where the photon is, if you like, the chunk of energy that any light source might emit. And around the world, we've seen amazing advances in governments and other stakeholders pushing light right through to the frontier. Here are just two examples. The National Photonics Initiative in the United States, just approximately a month or so ago, pioneered a collaboration between industry, academia and government to raise the awareness. And the House Appropriations Committee there approved funding for next year in several areas in this area of photonics. In Europe, we've got the Horizon 2020 program coming to an end, and then Horizon Europe. And there, the photonics industry is ready to invest up to 100 billion euros to work with the European Commission in this new kind of public-private partnership to push the science and the agenda of light further forward. Now, I'm going to talk today about healthcare, but let me just show you how exciting light is. Most people, for example, here's one, have heard of the idea of detecting gravitational waves, billions of light years away from us. And this topic is in the news, it seems, every other week. But many people don't realize the manner in which we detect gravitational waves is actually based on light. And it's based on the idea of interference. So in the top right, you see the idea of the interference of water waves. And that interference pattern is extremely sensitive to the sources and the path that each of the sources propagates or moves along. And here you see these two arms of an interferometer that's going out in this V shape in the top right, which is essentially the same as you see in the bottom left. This incredible and unprecedented advance, really pushing our understanding of the universe, was the winner of the Nobel Prize in Physics in 2017, and light was right at the heart of making that amazing measurement and has opened up a whole new field for scientists and wonderment for young and old alike right now. We also, believe it or not, can exert forces with light. If you enjoy a glass of wine sometime, look up, hold your wine glass up to the light, and look at the glass light coming through the stem. As it comes through the stem, the light can be twisted because the stem is usually some kind of ground glass or shaped glass. And that bending or scattering of light as it goes through the stem, believe it or not, exerts a minuscule force, if you like, on the stem, but it's so small that we can't see it and use it. However, scientists over the last few decades have used light in a more attractive way as a microscopic tractor beam to hold cells, as we see in the bottom left. Work at St Andrews holding simple red blood cells to look at disease for the immune system. We can actually take a single molecule, like a piece of DNA, and we can move and crush it and move it around. And this exciting idea can be used to exert forces and understand actually what happens in critical diseases such as motor neuron disease. So people can study kinesin motion in our cells, actin, myosin, and so forth. Again, this exciting idea was contributed half the Nobel Prize in Physics just 2 years ago, again emphasizing the importance and excitement that light brings to the world around us. So I'm at St Andrews in the North Hall, and here working in physics and also with laboratories both in medicine, and it's a pleasure to have the School of Medicine have a seamless interaction with physics through a continuous link, which means we can avoid the Scottish weather when students are moving back and forth. But it's a pleasure to have laboratories both in physics and in medicine, and also to be part of initiatives such as the James Mackenzie Early Diagnosis Centre and the Centre of Biophotonics, which are now integral parts and bringing people together from the whole university, looking at the area of biophotonics. And biophotonics means our science of light applied to the biomedical and biological world. The exciting thing is we're not just restricted to biology and medicine. Physics, we've got other disciplines very close by, which makes this an outstanding environment to perform excellent science, which is what St Andrews is really now renowned for, and it's a pleasure to be part of that. So one of the things that we've looked at in the last few years is nephrotic disease. So we've been targeting in my group the science of light and then using it for really specific diseases. Now this is something where your protein in the urine and your low blood albumin levels and swelling can occur, and the degree of protein in the urine can maybe make a huge difference. And here, that loss of protein leads to low protein levels in the blood, which causes tissue to draw in water. Now this means that in nephrotic or kidney disease, as you will, you can see the structure of the cells changing, and you can see some pictures of that here, and these sort of cell and structures actually change. But the problem is, to observe these for a pathologist is very difficult when he or she takes a biopsy. So we intervened here, and here by working with collaborators Mike Pristowski and Jim Pullman and colleagues at the Montefiore School of Medicine, at the Albert Einstein School and College of Medicine in New York, we developed a microscope system to actually look at this. So you can see that in the top left. So the green pictures are immunostaining, stained pieces of tissue. We can see these twisted or curved structures or foot processes, they're so termed. And what we did was we used a microscope to look at that. And then what happened was we then compared that with what is the gold standard, if you like, in this field, which is electron microscopy. And that's the picture on the bottom right. Now, the electron microscope is very powerful, but it takes up to a week to acquire the data, and the device is at least a meter and a half high, and extremely expensive. By using a form of fringe patterns, or patterns of light, as you can see in the middle there, we were able to get very high resolution and structure. So one of the exciting things for light is it can actually now approach the structure that we would normally associate with other methods in physics. And by doing that together, we managed to help push forward a new form of diagnosis for nephrotic disease, which has excited Jim, Mike, and other colleagues around the world. What are the challenges for light, and why can't we do more? Light is all around us. It's used in hospitals. It's used, of course, in many other things in our everyday life. But the problem is, just like when you hold up a light source to your hand, we all know that the light doesn't penetrate. It doesn't go very deep. So it's incredible that it's already been able to yield so much information, because light is so exquisite as a measurement device. And that's more or less the reason we've been able to be so successful detecting gravitational waves. But we would like to use it to actually understand disease more carefully. So to do that, what we're looking at now in St Andrews is new ways. So instead of looking at the surface, as you see in this picture here, just on the surface here, we're trying to look at new methods to go volumetric, to get imaging at depth, to get a proper 3D image of tissue. This is not our work, but other people are also looking at this area, showing it's an exciting venture. And here you can see the idea of detecting tumours with optical imaging. This, I think, is going to be one of the most exciting, novel, and profound changes that we're going to see in the next coming decades using the beauty of light. So how are we tackling this at St Andrews? Well, what we've done is we've tried to go deeper by, if you like, thinking about light and its property. It's a wave, just like waves you see at the beach. Here is a picture for waves on the beach at Israel. And you can see as the waves hit these barriers, they diffract or they spread out in curved geometries. And you can see that there. But what happens is as they spread, it means that the resolution we get or the information we can get from depth deteriorates. So what we've done is used light beams, as you can see in the right here, which are special patterns of interference of light. And when we use them, what we can do is retain that very good quality imaging and allow the light to go a little deeper than it normally should, to allow us to actually gain a lot more information. So we did this, and we developed a beautiful device in collaboration with our funding bodies, and then this work has recently been commercialized by the company in Scotland, M Squared Lasers. And these are examples of work from other laboratories using our very same principles as we see at St Andrews. So here on the left, you're seeing basically parts of the mouse brain imaged at a 1 micron resolution. A micron is 1,000th of a millimeter. So the resolution is incredible that we can actually see there. Here we have another specimen. It's not a banana, it's actually a worm called C. elegans, which is used to study neuroscience and neuroscience diseases, again taken this time at the London Institute of Medical Sciences. And these devices are now in over 20 laboratories around the world doing a variety of techniques. But what the beautiful thing here is we can take really exciting deep images, and as you can see, as we zoom in, the thing that we couldn't really do very easily before is now achievable and in our hands. We can go deeper. So you can still see the structure inside that worm, yet you can see the whole structure of it as well, the global size of it too. This is something that's another group in Sweden are studying. This is actually looking at the gut organoid, and they're looking at colon disease. And again, you can see the beauty of the structure here, that in this mouse intestine model, they can see nuclei, stem cells, and paneth cells. Again, the beauty is they can render a beautiful three-dimensional image that is actually quite unprecedented for them. And we're very excited to be able to help them to understand this and actually help us in looking at diseases that ultimately are affecting the quality of life for so many around the world. So we'll just wait, and this will zoom in, and you'll see again the beautiful structure that the scientists there can capture to help them understand exactly how the cellular processes are changing in the presence of drugs and other conditions. Here we go, zooming right in there, and you can see that we still retain incredible depth and high quality. So we're very excited that this work not only has been a great success for the science of light at St Andrews and a commercial product, but also won an innovation award in the Institute of Physics 3 years ago. We're now moving towards new geometries and ideas. Here in the right, what we're doing is we're using, instead of a camera to actually record the images, we're using a point detector, and we're projecting patterns rather like chessboards or checkerboard patterns of light into the tissue and then collecting the light out. And this method, because we focus in this way rather than we focusing in space as we do normally, we actually use a method in focusing in time. And this was just 2 years ago, but we managed to get information for hidden objects, and we're now working down to millimeters and beyond in tissue. And we're very excited about this new innovation developed at St Andrews that will help even the method things you've seen before go even further and get higher clarity of imaging. Light scattering in itself is very important, and the methods I've shown you so far rely on what we call fluorescence. Light comes in, and usually there's some kind of biological marker that gives us some kind of stain or color that we can actually record the biological system. But of course there's other methods. C.V. Raman, the Indian Nobel Prize winner, on a trip to Europe was inspired by the opulent colors of the Mediterranean and went back and understood Raman spectroscopy, where light shines on any molecule in the world and it shifts very slightly in energy. And that shift is indicative of the molecular bonds and the vibrations that are occurring. And this can be used in a number of ways. Now we're using it here again in the middle, as you can see, to look at cervical and other cancers. This work is, for example, work we've done again at Albert Einstein Institute of Medicine in New York in the United States. But we've used this also for lung cancer and bladder cancer. Most recently, we tackled the idea of bacteria, which is obviously something that is very topical at the moment. And we looked particularly at TB. This was work in conjunction with Stephen Gillespie's group in the School of Medicine. TB is an ancient disease, a global health problem, and is not going away. And in fact, is rising in many, many parts of the world. It's the 9th leading cause of death worldwide, with over 1 million deaths in 2016. Now, what happens with TB is that we can actually develop antibiotics to help treat this, but what we don't know is how those antibiotics stop working or what occurs during relapse. So we need to understand something about what happens to the bacteria in this process. Here you see TB deaths in the European Union. And you can see how many there are already in 2015. It's an area of real concern and something we need to tackle in the very near future. So what we did very recently, just this year, is developed a new method to use sound and light together to study TB. So believe it or not, sound waves, just like light waves, can grab and hold particles, just like we saw the force of light earlier. Here the sound waves in the top left, rather like the sound waves you might get from a musical greetings card, can actually grab those little black bacteria and confine them in the center. We developed this little microchip you see here, which is just 1 square centimeter in size. And there we could look at compounds such as lipids, carbohydrates, and others inside the bacteria and actually understand what was going on. The beautiful thing is we used an antibiotic here, and we could correlate the behavior of the bacteria cells depending on what the lipid content was, which again was a world first for us and exciting work in conjunction with Stephen Gillespie and Public Health England. This has been a very promising result that we hope, using this Raman idea here to collect the molecular signature of the lipids, that we want to push forward and see where that can go. It's not all biomedicine. Food security is something that we're very excited about and is very important. Here is a story showing that the Scotch whisky industry is a multi-billion-pound export industry and is it the water of life. But of course, whether you're a whisky drinker or not, there's counterfeit whisky out there, and there's also other drinks that actually people duplicate or counterfeit, which puts people's health at risk at many levels. This was something we wanted to attack. So using absolutely the same principles of small chips, using the Raman and the fluorescence ideas, we developed a beautiful little method a few years ago to look at whiskey.
I'm going to take a sample of A.
Okay.
Okay, now you take a sample, baby. Don't use the traditional method. There's a spittoon somewhere.
I'm not going to spit it. We're nearly at the end of the show. It's going to be fine.
So, Dara, you can taste that, see if you know what type of whiskey it is. And over here, the computer is going to have a look at this sample I've taken.
It's a nice whiskey. Glenmorangie. Right, that's... So we run the light to it.
And the computer comparing it says it's a pure malt, correct, but a Japanese pure malt.
Okay, so let's find out if the computer is right, if I'm right. Is the computer right? Yes, it is. Very good. Well done.
So that was a demonstration of our device, and a very stressful day, I must say, with everybody in the studio watching and looking at what we're doing. Excuse me. It's been a great pleasure to discuss the science of light, of course today, but also in many surroundings and international meetings with real stakeholders and right through to governments and even with the royal family here. So it's been a great pleasure to do that. And I think this is something that's very exciting for science, to communicate why we're doing things and the importance of what we're doing is making a real difference to human health and fundamental discovery. So I hope I can continue doing that with the ideas you've seen today and pushing further, both for the advancement of science and also at St Andrews as well in the coming years. So I'm going to stop there. I'd like to acknowledge all members of my group who've contributed to the work and these funding bodies. 2015 was the International Year of Light. And there, Ban Ki-moon says science and light science is a discipline that cuts through in the 21st century that can actually address global challenges. I couldn't agree more. I hope in this short presentation you've seen some exciting and interesting things that have inspired you to think about light perhaps in a different way. And I'll be pleased to talk with any one of you about anything that you've heard or even not heard where light, you might be curious, makes a difference in our everyday surroundings. So I'd like to stop there and thank you all for your very kind attention. Thank you.